My name is Nonjamiso Mazimsimanga, and I am one of the research coordinators at VIAD, and I have the pleasure of announcing our program to you. I hope you've each got your copy of the program so you can also follow where we are. And I'm going to begin by saying thank you to all of you for joining us at this VIAD event. This is quite an auspicious one because we get to inaugurate the incredible Professor Oron Catts, and we are quite excited about it. I won't say much because if I do, I'll just go on and on. And I'm going to very happily begin by introducing our wonderful respondent before our Professor Leora Faber, the director, introduces Oron with his incredible biography. So, just to bring you into who our respondent is, Professor Stacey Hardy is a writer, researcher, and editor whose work explores the intersections of embodiment, the individual, and society. Stacey Hardy has written and appeared in various anthologies and journals, including the New Orleans Review, New Contrast, the Evergreen Review, Black Sun Literature, and more. She is the author of the short fiction collections Because the Night and An Archaeology of Holes, which is my personal favorite. Her critically acclaimed plays and award-winning librettos have been performed globally at venues and festivals such as the in Provence. Hey, Provence. Thank you, Sadie. The Aix-en-Provence Festival in France and the Royal Opera House in London. Stacey Hardy is also lecturer in creative writing at Wits University and an editor at the Pan-African Platform, Chimurenga, a visiting fellow at the Newfound Collegium at the University of Chicago, and a partner in the African Creative Writing Initiative, Saseni, and a founder of Ubutula, a project that develops new writing from and against gender-based violence. Stacey Hardy has hosted interdisciplinary workshops, writing intensives in numerous locations, including Dakar, Kigali, Cairo, Tombwa in Angola, Nairobi, Lagos, Berlin, New York, and many more, and online, for those of you online. Additionally, she has facilitated graduate seminars and writing workshops worldwide. Please, can we give a round of applause for Stacey Hardy? We're going to begin with an introduction by my colleague and really amazing curator, Brenton Mott, who runs the BioArt and Designs Africa Strand. Brenton.
Hello, everyone. Let's get some audience participation. Some verbal interjection might be in order. So, thanks, Nonabisa, and thanks to Leora for asking me to talk about a topic which is very close to my heart, which over the past couple of years has become something of a sort of passion and a life's work, if that's not too dramatic. So Leora's asked me to talk about the research strand that I curate called BioArt and Design Africa. So I tend to get over enthusiastic and forget about making the most salient points. So I've taken the liberty of writing some notes. So I'm going to be referring to these. And so I'll just start. All right. And welcome to the latecomers. All right. So there are very many understandings and definitions and views and opinions of what art-science collaborations might look like, and more specific to our work, what bioart and biodesign might be. This is because bioart is a fluid, shifting term that cannot be pinned down, particularly as the creative work produced constantly changes in response to developing biotechnologies, changing biomaterialities, and scientific practices. However, a common understanding of the term bioart is that it involves practices that deal with the hands-on application of the life sciences or biotechnologies. Implicit within this art form is the physical use of biological matter as art-making media using processes known as wet biological practices. Artworks may include living and non-living biomatter, such as cells, extracts of cells, tissues, DNA extracted from life itself, bacteria, fungi, plants, spinous invertebrates, hard-bodied insects, and necessarily controversially, sometimes entire mammals. This is part of the working definition of bioart and design that we use in the VIAD research stream that we've named Bioart and Design Africa. Bioart and Design Africa, which we lovingly shorten to BARDA, is a creative space of play where art and design practices converge with the life sciences through the use of biomatter as medium and content using scientific equipment, protocols, and procedures. Departing from the historical dichotomy between creative and scientific disciplines, this space fosters transdisciplinary research where artists, designers, and life scientists come together on an equal footing to explore the creative and theoretical possibilities at the nexus of their fields. As such, BARDA offers fertile ground for introducing speculative methods of working with biomatter, engaging in novel forms of collaborative practice, exploring innovative ways of working with existing biomaterials and producing new ones, developing unconventional methodologies, and using practice-led research approaches to arrive at unprecedented outcomes. We embrace and nurture transdisciplinary critical and creative modes of engagement that catalyze innovative approaches to more than human ecologies and their relations, environments, and futures. So in a nutshell, it's about play, about constructive play is what we do. However, our use of the term bioart and design extends beyond wet biological practices to incorporate the diversity of the life sciences as they manifest in creative practice and visual representation. Our use of the term bioart and design is broad and generous. We consider biomatter at individual, community, geographic, cultural, economic, and planetary levels, while incorporating forms of bioart and design that involve the use of living and non-living organisms as media. The work we produce and our artists in association also includes other ecologically oriented forms of creative practice that are not bound by specific medium or methodology. Our position is well encapsulated by a definition that observes that bioart epitomizes a coalescence of art and science. It is an emerging contemporary art practice that uses a wide range of traditional artistic media interwoven with new artistic media that are biological in nature. This includes molecules, genes, cells, tissues, organs, living organisms, ecological niches, landscapes, and ecosystems. In addition, bioart expands into conceptual art, using biological processes such as growth and evolution, cell division, photosynthesis, concepts about the origin of life and evolution, and explaining these abstract concepts as new artistic media. In this time of global challenges, bioart communicates thoughts and feelings that involve the relationship between the artist, the science, the public, the biological organism, and the biological concept. Under this overarching rubric, research done in BARDA is structured around 7 interdisciplinary focus areas. Each is underpinned by the core theme of decoloniality and entangled relations between the human and the more than human, and the social and climate justice. So I'll just briefly mention each of these. The first area we've termed microbial ecologies, and this focus area is realized through a unit that we've termed the Creative Microbiology Research Collab. The CMRC, which was founded by VIAD Director Leora Father and the Director of the Water and Health Research Center, Professor Tobias Barnard, who's sitting over there. These two pioneers at the University of Johannesburg created this interdisciplinary collaboration dedicated to producing and disseminating art and design research that interfaces with microbial science. Its focus is on the use of microbes, which is bacteria, viruses, components of these fungi, and related content to generate art and design outputs. And one of the first outputs of the CMRC was the custom-built laboratory at the Faculty of Art Design and Architecture, which is the home of this focus area. Next is ecological practices, or traditionally known as eco-art, which we have termed reparative and precarious ecosystems. This is a contemporary form of environmental art which is related to local and global environmental concerns. Artists and designers use ethical practices, which itself is a broad term and open and subjective, use ethical practice to redress how human intervention impacts on ecospheres. The work can involve ecological systems restoration, as well as socially engaged, activist and community-based interventions geared towards achieving social and climate justice. Third, botanical ecologies is the focus area dedicated to developing approaches using plant science, plant life matter, and related biotechnologies that foster relationships between the human and the botanical world, and between plants and their biotic environments. Fourth is biomedical ecologies, an interdisciplinary focus area that draws on arts, humanities, and medical sciences to shift westernized and patriarchal powers and politics of disease. Topics of exploration include histories and cultures of medicine, disability studies, gender and the body, communities in crisis, bioethics, and public health. Work produced under this focus area may deal with entangled relationships between the human body, bacteria and viruses, host-pathogen interactions, contagion and contamination, immunology, and the body in relation to the medical industrial complex. Fifth, material ecologies incorporates nature-centric and living design, in which biomatter and ecosystems are essential components in new synthesized design forms. It covers design that investigates the development of a product through the invention of new biomaterials or the repurposing of existing ones, new production processes, as well as sustainable development options for creative and functional applications. It includes work that replaces industrial or mechanical systems with biological processes, and work that also goes beyond functional or speculative design into the realm of visual art. Sixth is queer ecologies, which brings bioart and design together with queer theory and ecology to shift paradigms away from the binary, rigid, and heteronormative ways of understanding sexuality and gender. These intersectional and interdisciplinary practices break down the assumed heterosexuality and cisgenderness in biology that has been the baseline for scientific studies for centuries. The inherent non-sexual queerness of fauna and flora is drawn upon to prompt sexual and gendered interdependency and fluidity. Finally, the seventh focus area of our work is Afrofuturist ecologies, which, when linked to bioart and design, centers ancestral knowledge, technological innovation, deep time, and speculative imagination. Challenging Eurocentric scientific narratives, this area embraces relationality, multi-species coexistence, and ecological repair through African biomimetic traditions. By merging storytelling, ritual, and biotechnoscience, these fields generate new political, aesthetic, and biological possibilities for decolonial futures. Although bioart and design practice has gained traction in the international arena since the 1990s, its recognition in South Africa and the broader African context remains limited, thus presenting a unique opportunity for creative and theoretical exploration. While taking cognizance of work done in the global north and drawing from it, BARDA aims to generate insights and perspectives grounded in African contexts. It is founded on the conviction that work produced under our expanded definition of bioart and biodesign holds enormous transformational potential to resituate the site of knowledge production and generation. As many decolonial and anti-colonial scholars argue, the key issue underpinning the racial capitalist scene is justice for the billions of Black and Indigenous lives that have been commodified, quantified, and extracted using a geological approach to ecosystems, to knowledge, and to relations. Achieving climate justice as a form of social justice necessitates shifting the logic of capitalist colonial science towards indigenous and post-biological ontologies. In this regard, BARDA acts as an incubator for a form of decolonial practice which has the potential to change the dynamics of Western knowledge systems linked to power and control. These include those hegemonic forces that have shaped relationships with humans and the more than human, with living and non-living matter, with nature and culture, and with life and with death. The work we do recognizes the ability of bioart and biodesign to be mobilized as an agent of political change and transformation in relation to issues around race, gender, and class. And in conclusion, by fostering experiential approaches to what Karen Barrett terms interactions based on empathy, care, and respect, BARDA promotes symbiotic entanglement and interdependence along with the multiple forms of life that exist in trans-species alliance based on diversity, complexity, and hybridization. We believe that the combination of alternative forms of collaborative practice, experimental ways of working with biomatter and biomaterials, unconventional methodologies, and practice-led research with indigenous forms of knowledge, which foreground ecocentrism, that's a very long sentence, could generate a powerful platform to erode the violent histories of slavery, humanist thinking, imperialism, colonial oppression, and the racial capitalist scene, and expose their roles as agents of destruction in relation to the ecosphere. The last sentence, also a long sentence, to the production of insights and perspectives that arise from and pertain to an African context. BioArt and Design Africa, the research strand, provides a critical Africanized footprint, where bioart and biodesign become a means to prompt the transformation of radical, ecocentric, decolonial practice. Thank you.
Professor Leora Faber, Director of VIAD Research Center, will now introduce Professor Oron Catts. Briefly.
Good evening, everybody, and thank you for joining us for this very auspicious evening in which I have the pleasure of welcoming Prof Oron Catts as a visiting professor in the Visual Identities in Art and Design Research Center. We're enormously proud and extremely excited to welcome him as a BP, and are very excited about the possibilities of working with him and the center, which maybe he will tell you a little bit about, the new Symbiotica Center, which is developing in Perth at the moment. Associate Professor Oron Catts is an artist, researcher, designer, and curator, and a world-renowned expert in the field of bioart and design. He was recently appointed academic lead and director of the Institute of Advanced Studies at the University of Western Australia. Prior to this appointment, he was the director of Symbiotica Centre of Excellence in Biological Arts, School of Human Sciences at University of Western Australia, which he founded in 2000. And what is particularly remarkable is that Symbiotica was the first research laboratory of its kind, enabling artists and researchers to engage in wet biological practices in a biological science department. The center enabled a pioneering means of creative practice where artists and designers actively use the tools and technologies of science, and specifically biotechnology, to explore the creative possibilities of art-science collaborations. The center offered a Master of Biological Arts, a coursework and dissertation. And over the 2 decades, Symbiotica has hosted more than 100 artists as residents, including influential figures in the worlds of art and technology, such as Oron, Steve Kurtz, Paul Van Koos, Kira O'Reilly, and Lou Young. Under Catts' leadership, Symbiotica won the Pre-Arts Electronica Golden Nika Hybrid Art Award in 2007 and the Western Australian Premier Science Award in 2008. Together with Ernat Zur, Catts founded the Transformative Tissue and Art Culture Project, TCNA, in 1996. This project is considered to have been a groundbreaking endeavor in the then newly emergent field of bioart. It explores how tissue engineering can be used as a medium for artistic expression, based on the use of regenerative biology techniques to create semi-living art using cells, tissues, and technological surrogate bodies. From 1996 to date, the TCNA project has worked internationally to produce artworks, curated exhibitions, performances, and publications that focus on the impact of tissue engineering on society. While the TCNA project is considered a pioneering collaboration in the field of bioart, the impact of Catts's personal work extends beyond art-making practices and is often cited as an inspiration in diverse areas such as lab-grown food, tissue culture clothing, semi-living sculptures, new materials, textiles, design, and architecture. Catts's work is also considered as a precursor to the cellular agriculture and biofabrication industry. Catts was a research fellow at the Tissue Engineering and Organ Fabrication Laboratory at Harvard Medical School, Massachusetts, in Boston from 2000 to 2001, a visiting scholar at the Department of Art and Art History, Stanford University, a visiting professor of design interaction from 2009 to 2012, and professor at large in contestable design from 2015 to 2017 at the Royal College of Arts, London. In 2012 to 2013, he set up the Biophilia Art Lab, which is a dedicated laboratory for biological art and design at the School of Art, Design and Architecture at Aalto University, Helsinki, where he was a visiting professor. In 2009, Catts was recognized by Thames and Hudson's 60 innovators shaping our creative future. And this was the book in the category of Beyond Design, and also by Icon magazine in the UK as one of the top 20 designers, quote, making the future and transforming the way we work. Catts has curated over 12 international exhibitions, published widely in scholarly journals and edited volumes, and is exhibited in prestigious venues such as the Centre Pompidou in Paris, MoMA in New York, the Mori Art Museum in Japan, Arts Electronica in Austria, and the National Art Museum of China, amongst others. Catts' stature as a pioneer in the field of bioart, the founder of the TCNA project, and his position as a leading global bioartist, and 20 years of experience as director of Symbiotica, as well as his current position as academic lead and director of the Institute of Advanced Studies at UWA, speak to the enormous contribution that he could make to the growth and development of BARDA. His wealth of expertise in the field of bioart and design opens significant potential for expanding and deepening BARDA's current research. And it's with that I have great pleasure in giving you over to Professor Prof Catts.
Thank you so much, Oron. So great to be here, and thanks for this really expensive introduction. Hopefully I would live up to the expectations. Can we get the presentation up, please? Yeah, I'm so happy to be here, and thanks for VIAD team for inviting me, and Breton and Liora. I spent a few days, my first time in South Africa. Hopefully I'll come back here for longer. And we have to sort out the technology. It worked before, I can promise you. But this is actually a really great opportunity to talk to you about trying to control systems that existed outside of our own influence. So the issue is that we live in a really interesting time. Yeah? We live in a time where we're trying to make technology more lifelike, but obviously technology seems to resist. So basically our technologies are becoming so complex that we are already recognizing the fact that we are not controlling them very well. At the very same time, we're trying to control living biological systems as if they're a piece of technology. And really, that will be most of what I'm going to discuss with you today. I titled the talk "Life is not what it used to mean," because I really want to talk to you about how a relationship to this idea of life is changing and shifting. But before I start, I would really like to acknowledge the fact that I'm working from the lens of the Whadjuk people of the Noongar Nation, so the indigenous and traditional owners of the land for which I'm living and working. Some of you might know this place as Perth, Western Australia, and for the indigenous people it's called Boorloo. And so I would like to acknowledge the fact that I'm working for them and pay my respect to elders past, present, and emerging, and acknowledge the fact that the land from which I'm working was never ceded. "30 years ago, this image hit the media. Many of you here are born into a world that mice have human ears growing from their back. I wasn't. When I saw this image, I was taken late and felt that I need to somehow deal with it. And this is a work that was done by scientists using a technology called tissue engineering, which I'll describe shortly. But for me as an artist and a designer, it represented something else. It represented the surrealist dream coming alive. It represented a way in which humans reach a stage where something that appears in all human cultures, this human-animal mix, the human-animal chimera, was moved from the realm of fantasy and magic to the realm of reality. This was also represented something that some artists aspire to since the beginning of time, and that's to sculpt with the very material of life itself. I was so taken by that I decided to explore it further. I actually ended up, as you heard in the introduction, I ended up in the tissue engineering and organ fabrication laboratory in Harvard Medical School, where the main person who developed this technology basically worked from. So the way I decided to investigate it further is just going into the place that I found the most disturbing, in a sense, where this thing happened, to understand how it happened, what it means, and how I can actually work with it. The idea of tissue engineering came in the early 90s, late 80s, with the notion that our understanding of how to fix bodies was lacking. So the blue slide there on your left shows how we imagined we're going to repair bodies in 1989. What you're seeing there is what I refer to as the Tillman approach, the notion that if we are missing a body part or if we need to replace a body part, we would just use the dry and hard electrical and mechanical engineering devices and bit by bit replace the body with those devices and transform us into Tillmans. The problem is that the hard, dry technologies of electrical and mechanical engineering are not very compatible with the very moist, corrosive, and self-repairing environment of the body. So in the late 80s, early 90s, something else happened. Scientists started to think about ways in which we can harness those latent regenerative powers of the body to basically transform the body to its own technology of repair. The idea was that we can somehow be able to grow body spare parts or parts of bodies outside of the body and implant them into the body in what seemed to be a very simple system. So you start with a three-dimensional biodegradable polymer, which you see up there in the top. So basically, scientists discovered materials that are compatible with the human body, that can break down as tissue grows over them, and basically, bit by bit, you replace this artificial material, the artificial construct, with living biological tissue. A bit like a lost wax technique if you do jewelry. Yeah. So you sculpt, basically, physically sculpt that organ out of the polymer, and you then seed it with the appropriate cells of the organ you're trying to replace. That was before they worked with stem cells. Now with stem cells, it's supposed to be simpler. You grow it outside of the body in vitro. until the cells kind of take shape. And then you implant it in the body and you're supposed to have a new organ. In theory, it sounds great. More than 40 years after this technology started, we still don't really have many organs that are grown in such a way. No, don't wait for organs waiting for you on a shelf in a hospital somewhere if you need a new organ. You know, we still have to rely on organ transplant and many other technologies that are trying to help us to repair bodies. But tissue engineering in this form is not really working very well, and it might not ever work. But as with many other technologies, there's maybe other uses for it. And this is what I was investigating since the mid-90s. So my interest was what happens if we can actually grow a three-dimensional functional organ or tissue and never put it back into the body. So back in the 90s, there was an international consortium of scientists that proposed that in 10 years' time, they'll be able to grow a fully functioning heart outside of the body. We're not there, again, way over the deadline. There's no hearts that are growing in such a way. But my question was, if we can grow a heart, why not grow pumps? But then I was also very disturbed by this proposition. What does it mean to start to think about the body in such a way that we can start to grow those products that are actually functional living tissues? And that became kind of my pursuit and my interest. So I decided to go into a lab. Actually, the very first lab I went to work in was an eye research institute. The scientist that I worked with was trying to develop an artificial cornea. So he wanted to see how the skin that covers the eye would engage or would react to the materials that he was developing. So a bit of a trigger warning. The next image presents a dead animal, with some blood, and if you find it disturbing, just shut your eyes. So that's what we were confronted when we arrived to that lab. And the lab would receive a cardboard box full of half rabbit heads twice a week around lunchtime. We were told the rabbits were killed for food. The heads were then cut off and sent to a brain research institute, where they would cut the head in half, take the brains out, and then send those half rabbit's heads to the lab where I worked in. We learned the technique of how to pull the eyes out, and then we would put the eyes in antibiotic solution. You see the red liquid there in the vial, and leave the eyes there overnight. So some of you might not get this reference, but that was actually the reference that we got when we entered the lab for the first time. So those of you who don't know, this is from Blade Runner, the original. And as we entered the lab for the very first time, a postdoc was running with a vial of eyes and quoting, using this very same quote, eyes, eyes, all I do is eyes. I'm a genetic designer. And we knew we arrived to the right place. At least we started to have shared metaphors in regard to what's going on. So we would have a conversation with the scientists, we would look down the microscope, start taking those bits of skin from the eyes, as I said, more than 24 hours after the animal itself was dead. And then we would start culturing those cells and have living cells. And for us, it was quite confronting, because you can't be more dead than being a half head with no brains sitting in a cardboard box. But we still were able to isolate living biological material from something that was dead for more than 24 hours. That started to hit us. So I was working with my collaborator, Yonat. So when I say us, it's often me and Yonat. So it basically hit us that we were lost for words, that the language that we used to describe our encounters wasn't relevant to this experience that we had in the lab. And it's kind of important, as Simone Weil reminds us, when a word is properly defined, it helps us to grasp some of the concrete realities or concrete objectives of methods or of activities. To clarify thought, to discredit the intrinsically meaningless words, and to define the use of others by precise analysis. To do this, strange though as it may appear, might be a way to saving human lives or any other lives. Yeah, so this idea that we are now going into labs and there's things that we don't have to describe, and encountering things that we have no words to describe, might be actually quite important for us to try and develop a new language around it. The idea that life becomes an engineering, or biology becomes engineering and life becomes a raw material, is not that new. As early as 1895, A.G. Wells already recognized that, and he wrote, "We overlook only too often the fact that a living being may also be regarded as a raw material, as something plastic, something that may be shaped and altered." And obviously he was an author, he was a man of words. He then went to write The Island of Dr. Moreau, those of you who know it. We're trying to kind of engage with this notion that we're starting to treat and think about living beings as a raw material for us to extract value from. We also, as early as 1913, when this technology of tissue culture, when this realization that you can somehow take cells and tissues from a complex being, complex organism, and keep them alive for long durations of time, was actually quite a sensational thing in the media. And this is a quote from one of the very first scientists that was working with tissue culture in the early 20th century. And he said, "It seems rather surprising that recent work upon the survival of small pieces of tissue and the growth and differentiation outside of the parent body should have attracted so much attention." So humans already been engaging with those kind of conversations, but we can account it for it by the way the individuality of an organism as a whole overshadow in our minds the less obvious fact that each one of us may be resolved into a myriad of cellular units with some definite structures and with autonomous powers. So this idea that thinking about our own bodies, thinking about what it means to have a body, is starting to change and shift as we start to recognize that we are actually a community, a collection of cells that can exist independently from each other, obviously with a lot of technological support. And that was 1913. Those conversations happened, but we forgot about it. One of the issues that I keep on encountering with biology is that we are suffering from acute amnesia or cultural amnesia. Those stories, we told ourselves those stories before. Some of the issues with those stories, they led to a really bad ending. Some of the people who were involved heavily with tissue culture and really the beginning of tissue engineering in the 1920s and 30s ended up being Nazi collaborators. They were wiped out of history. So rather than learning the lesson, we are doomed to repeat it. And we're now telling ourselves very similar stories to what we told ourselves 100 years ago. The idea of putting bodies and machines together also created some really strange things. So this is the first premature baby wards in the United States. So basically babies in incubators. It wasn't in a hospital. This one was in the Pan American Expo in Buffalo in upstate New York, but it wasn't in the place where they were showing off new technologies. It was in the place of oddities. And we know it because after this exhibition or the expo was done, they moved this whole installation to Coney Island, to the fairgrounds. And you might be able to see it says infant incubators with living infants. The reason for that, and there's many reasons, but what I would like to focus on is the fact that there was no cultural place to put human bodies and machines. There was no place to recognize them as entities that needs care and attention beside the freak show. And they actually had to pay their way in. So the only way those babies could survive was to basically engage with humans' curiosity and interest in the freak in order for people to pay money so those babies can survive. And there was about 80% success rate of those incubators, as opposed to almost zero if those babies would be left unattended. But still, it took many, many years for the incubators to actually be introduced to hospitals. And even for a while, there was this strange overlap where baby incubators were sold from Dreamland, Coney Island, to hospitals and amusement parks. And again, I would say that this has a lot to do with the fact that we had no language to even engage with the idea of tiny human babies that are being kept alive by machines. We took a while to basically move from the place of the freak to something which is societally acceptable. And to some extent, it's correct with this mouse. So going back to this mouse, because it was my muse. something really interesting happened. So first of all, the way the mouse was presented to the public back in 1995 was quite curious. This mouse lives on a petri dish. At least it was photographed on a petri dish, and I suppose we all know that mice don't really grow in petri dishes. But that was a very deliberate decision of the BBC documentary crew to stage the mouse on that dish, because the petri dish becomes the new stage in which the theater of life is now performed. One of the scientists, there's a really long story, I won't go into it, but one of the scientists that was involved with it, and he actually showed the mouse to the BBC documentary crew against the advice of his colleagues, then took ownership over it. And when he realized the huge public interest in that, in the freak, he did something which was quite curious. He cut the ear from the back of this specific mouse, because obviously they had quite a few other mice, and cast it in resin as a cultural artifact, as a museum-ready specimen. You can't gain any scientific data from that, but that wasn't his interest. He realized that the importance of this ear is, as a cultural object, is much more significant than the importance of that as a scientific object. In 11 years after that happened, he did something even more curious. So in 2006, he decided to copyright any depiction in any media of a mouse that have a human ear on its back. He felt that he owns this piece of the public imaginary, and he's the only one who will be allowing us or disallowing us from propagating it. So he put a sculpture, a photograph, and a drawing, and all of it, and he thought that he might be able to control how this image is going to be distributed, how culture is going to deal and respond to that. This is not something, this is not something scientists often do, yeah? This idea that suddenly they realize that actually they are more engaged in culture than in science itself. Another example, which is kind of a bit harder to talk about in terms of the freak, is Dolly the sheep. So Dolly the sheep was a technological feat. Dolly was the very first mammal that was cloned. And after Dolly died, the Rosalind Institute, where Dolly was developed, decided to donate the body of Dolly to the National Museum of Scotland in Edinburgh. And the people in the museum there were in a bit of a conundrum. The museum has a natural history wing, and obviously this is where Dolly was treated and stuffed. But Dolly's place wasn't in natural history. Dolly was a product of technology. Dolly is technology. So this image is really important for me because this is kind of where I operate. It's Dolly being moved from the natural history section of the museum to the technology and society and culture section of the museum, the man-made, the human-made part of the museum. This is kind of the realm where I would like to believe that a lot of the art that is engaging with the manipulation of life kind of exists. Now, many museums wanted a piece of Dolly. Some museums have sweaters that were knitted using Dolly's wool. But one day when I went to the Science Museum in London, I encountered something quite curious. Some curator decided it's worthwhile to collect Dolly's droppings. We can fetishize, we can start to worship Dolly's droppings. And again, notice on what stage they are. They're in a petri dish. I don't think any of us like the idea of having droppings in a petri dish, but that's how it's being displayed. But as we say, you know, history belongs to the winners. As far as I know, no museum collected any of the 277 failed attempts before Dolly was born. Yeah. So there's a very skewed idea in regard to how we present our technological successes or how we present life and technology. So now I move into some of the projects that I've been doing in response to that. So in June 2000, there was a big celebration across the pond, so in America and in England or the UK, where they declared the Human Genome Project complete. So the Human Genome Project was an attempt, and one of the most expensive scientific endeavors to try and map the DNA that we have in our cells and basically the genome, with the idea that somehow if we'll be able to read our genome, we'll have the instruction manual or the blueprint for what it makes us humans. The celebration in June 2000 was, as I'll show you shortly, wasn't so much a scientific celebration, it was more of a PR exercise. The problem was that a private company started to map the human genome, and they did it much faster and much cheaper than the public one. The media's attention moved away from this narrative of we would find a cure for everything if we would solve this problem, or if we would be able to read the human genome, to questions around what does it mean when private enterprise is starting to own part of us. Because what this company did, a company called Celera, as they were trying to find useful genes, they started to patent them as well. So the media was like, basically attacking the public, or part of them were attacking the public Human Genome Project, saying, why did you waste all of this money when a private company can do it so much better? Other parts of the media were going on, do we ever allow the ownership of who we are? But all were engaged in a very hyperbole discussion. All of them were talking about this Human Genome Project as the biggest thing since the discovery of fire. They were one of the proponents of this project basically said in June 2000, now that the Human Genome Project is complete, which wasn't, the impossible is possible. So we decided to see what shape those flying pigs would have. Now the impossible is possible, so pigs could fly. So we looked at a three-evolutionary solution of flight in vertebrates. So we know that we have birds, we have bats, and we have dinosaurs, or the pterodactyls. In Western iconography, usually if bat wings are attached to something which is not a bat, they'll be attached to devils and demons. Bird wings would be attached to angels. And dinosaurs' wings, or the pterodactyl wings, are not really attached to anything in our iconography because we just found out about them late in our history. So we said, okay, we wrote those three sets of wings out of pig tissue. We went through the whole process. I actually used, I designed those wings using a 3D software. We 3D printed them. We made them out of this special polymer, like in the case of the mouse with Oron's back. And we grew bone cells. Actually, we started with bone marrow stem cells that we then differentiated into bone, and we grew those bone structures made out of pig tissue in the shape of those three sets of wings. I think I said that. Yeah, we were invited to, that was a commission. And the commission came from actually one of the main funders of the Public Human Genome Project, an organization called the Wellcome Trust, that had a gallery at the time. When we proposed this project to them, they rejected it. And they rejected it in the most honest way possible. So if you want to get a rejection, at least try and piss off the people, because then they're honest. And the reason they rejected our work was because they said that it didn't represent the public opinion in regard to the Human Genome Project. And then they questioned both the scientific and artistic validity of our work. So when it comes to representing the public opinion, would you really go to an artist to represent the public opinion? Is that the role of artists? That's such a strange thing. So we responded and apologized that we didn't represent what they thought the public opinion is ought to be. And in regard to the scientific and artistic merits, this project was developed when we were research fellows in Harvard Medical School. We introduced through this project the whole area of three-dimensional computerized design and manufacturing of tissue constructs. They were then using it for trying to grow real organs. And the artistic merits, this work is currently, well, it's part of the permanent collection of MoMA in New York. So, sometimes it's good to be rejected because actually you can prove them wrong. But the strange thing is we started to exhibit, so we developed this project regardless, and we started to exhibit it around the world. And what we noticed is that in many cases, the galleries and museums that exhibited our work were kind of also implicated in this hyperbole rhetoric, to such an extent that people who came to see the work thought that they're going to see, if not flying pigs, they're going to see wings which are big enough for pigs to fly with. But those were tiny bits of wings, about 2 centimeters each, deliberately displayed in cheap jewelry boxes. And we kind of engage in what we refer to as the aesthetics of disappointment. So trying to kind of push against this hyperbole rhetoric. At the time, they were actually some of the biggest tissue-engineered construct ever to be grown, but people had the fantasy that somehow we can grow those large wings. So when it comes to the Human Genome Project, this is kind of just some, you can see Clinton and Craig Venter, who was running the private company, and the head of the NIH in America, the whole celebration, the whole rhetoric around it, yeah, which is basically the book of life, cracking the code, all of the stuff was going on. And shortly after, even the Wellcome Trust, even the people who rejected our work, the executive of the Wellcome Trust said, a few months ago, I compared the projects to the invention of the wheel. On reflection, more than that, but the code is the essence of mankind. But then they said, while the timing of the announcement may have been directed more by political than scientific criteria, blah, 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 blah. And then the joint announcements was probably more grandiose than the situation warranted. They admitted that they were hyping this up. Actually, now the Human Genome Project is considered to be a footnote rather than a major thing in the history of science. We recognize it as artists, but they were really offended that we shined the mirror back at them and told them where to stuff it. But this idea of the exaggeration is not just scientists. The people who are opposing those new developments in science would use the same type of exaggeration. The mouse with Oron spec, as I told you, was using a technology called tissue engineering, nothing to do with genetic engineering. It was actually very craft-based. You basically, a Chinese plastic surgeon was the one who crafted the ear out of these polymer scaffolds, and then it was seeded with cells. But when a big conglomerate of anti-science, if you like, or pro-environment, at the time they were quite incompatible, organization decided to have a full-page ad in the New York Times, anti-genetic engineering, they decided to use the mouse with Oron spec rather than a genetically engineered organism. Because as Dolly, who was cloned, which was getting closer to genetic engineering, she didn't look like nothing scary. They need to get something scary to freak us out. And since then, people still believe that we can somehow find the gene for human ear and put it in a mouse and somehow it will sprout on the back of it. So the exaggeration in regard to the power of those technologies is coming both ways, both from the people who oppose this technology and those who are trying to promote it. And as artists, we were trying to just figure out. We actually chose to go with the paradoxal wings. You know, we still need to figure it out. It's not that evil, but it's also not going to save us. Let's step back to 1951. 1951 was the time where the first human cell line was created. So when you take cells from organisms, most of the cells would have a finite amount of division they would go through before they would stop dividing. And that's one of the reasons why I'm getting gray and getting old. Our cells are getting tired as they divide. But there's one type of cell, well, actually there's a few types of cell, but one in particular that seem to lose this self-destruct mechanism, and that's cancer. In 1951, doctors from Johns Hopkins University Hospital isolated a cancer from an African-American woman called Henrietta Lacks and decided to see if they can culture those cells. Those cells, the cancer was so vigorous and that those cells just grew and never stopped growing. Those are what's now known as the HeLa cells. Those cells, obviously, poor Henrietta, she died of this cancer. But if you would collect all of the cells, those HeLa cells that were taken from her cancer that are still growing around the world now, we can fill up this building. There's tons and tons of those cells all over the world, distributed all over the world. What does it mean? There's a biomass of those cells far exceeding what it was when Henrietta was alive. Her family just found out about that quite late. And if you look for HeLa cells and the stories about it, you'll find some really interesting documentaries and books were written now. The HeLa cells were used for the development of the polio vaccine. They were sent into space. They're all over the place. But also, they contaminated a lot of experiments. Because those cells actually in the labs are considered to be weed. They would take over. If a scientist worked on those cells, very likely if some scientists would go and work on other cells in the same kind of working station in the sterile hood, the HeLa cells would jump in and contaminate it. To such an extent that 40 years after those cells have been developed, two scientists said, hey, those cells are not human cells anymore. They're a new type of microbe, a new type of microorganism, and we need to give them a new name. It's a new species. And so they basically say species originated in diverse ways. HeLa cells are the best known tissue cultured cells of human origin. We now propose in all seriousness, and they were serious, that they have become a separate species restricted to a particular environment, and that is the lab. So again, this language, yeah, are those cells human? Is it Henrietta? Are there something else? I used to run workshops where I would teach artists how to work with biotechnology. And what we would do is, like, we would work for a week with different types of organisms, and then the very last day of the workshop, I would ask the participants to devise a ritual to give thanks to the organisms that were part of this learning experience. And obviously, when it came to working with cells, and especially human cells, people tend to have special considerations towards them. So how are we going to, because we have to dispose of them by the end of the workshop, what are we going to do? How are we going to engage in some kind of a ritual around them? In one workshop in particular, we had this Greek artist, and as we were all talking and trying to find compassionate ways to commemorate Henrietta, ways to commemorate the contribution of those organisms to knowledge production and our specific quest for knowledge, this Greek artist stood up and said, a year ago, my husband died of cancer. In my eyes, those cells are not Henrietta's cells. In my eyes, those cells are the cancer that killed her. Killed the fuckers. Everything changed. The relationship to the cells changed. The whole kind of interest was shifted to somewhere different. And again, this kind of lack of language. Are there evil? Are there Henrietta? What kind of ways we need to devise in order to engage? And this is where we lost for words. So living fragments of biological bodies, forms of lab-grown life, requires a different epistemological and ontological understanding, and by extension, a different taxonomy of life. The liminality of this kind of technological approach to life can lead to a form of fetishism, which we, meaning Yonat and myself, started to refer to as neo-lifism. This fetishization of new approaches to life, and I'll expand on it shortly. We did a project where we were commissioned for another project, for an exhibition that was called HeLa, which was supposed to commemorate this whole story. So we decided to develop a piece which the title for the piece was actually taken from a 1972 book by Samuel Butler called Oron, which, if you have a chance to read it, it's an amazing book about technology. In some passage of the book, the guy is walking in a field and see a decaying sculpture, and he says, "For art is like a living being, better dead than dying." And in a sense, those HeLa cells are kind of perpetuating the death. Yeah, it might not be perpetuating the life of HeLa, of Henrietta. It's really perpetuating her death. So we decided to build a work around it. We used one of the most famous images of Henrietta, and we created, basically we used her silhouette, and we cut it out of this polymer that we were working with. And we then seeded it with those HeLa cells and put it in this device that was a life-giving device to start with. But in an indeterminate point of the exhibition, because we didn't maintain it, it became a death chamber. So this idea of the indeterminacy of what the cells are there Henrietta? Are there this new organism that was suggested? Are they alive? Are they continually dying? All of those questions were embodied in this one work that when the audience looked at it, they didn't know if they're seeing living cells or a death chamber. And that was kind of the importance of it. Just as a little anecdote of what happens when we try to show our work in museums, we were approached by the Centre de Pompidou in Paris for an exhibition called Designing for the Living. And as we were sending them proposals, they came back with the most amazing quote I ever got from a gallerist, where they said, "Unfortunately, I'm afraid that it would be difficult to realize a living installation work as part of our exhibition titled Designing for the Living. That's centered at George Pompidou, yeah? So we gave them this work, said, okay, you don't have to deal with this because we are going to have a dying piece rather than a living piece, and dying as a process, becoming dead, not totally dead. So the fetish of technological approaches to life overshadows the context in which life operates. I told you that we refer to it as neo-lifism. It seems that a biological milieu is transformed into an abstract technological instrument of control, where life is just another raw material to be engineered. Decontextualized life has been reconfigured, mixed and remixed, reappropriated and instrumentalized in such an extent that the technologically imagined potential of life stands for life itself. How long do I have? Yeah. 7 minutes. Okay, so I can just give you another project. One of our most famous projects, and one that actually appeared in the poster, so it will be remiss of me not to talk about it, is our victimless ladder project. So for the first few years of our practice, we were basically trying to grow sculptures that stood for different kind of symbolic gestures. Then in the year 2000, we actually grew meat, not because we thought it's a good idea, but because we thought this is kind of making sense. If we are engaging with the strange relationship we're forming with life, obviously you can't get more intimate with another life form than incorporating it into your own body. So what does it mean to grow or to eat meat that was grown in a dish rather than in the body of an animal? I won't have time to talk about that project, but that started a series of works that we refer to as the technologically mediated victimless utopia. And that was very ironic. The second project in the series made a lot of sense. If we grew meat, how about growing leather? So we decided to grow leather in the lab, but we decided that when we exhibit this work, we don't exhibit the piece of leather that we've grown, and we actually managed to grow leather that 20 odd years later, it still feels like leather. We decided to show a living or semi-living piece of leather grown into a shape of a jacket within a techno-scientific body. So also kind of in a sense reversing this idea of leather as a protective layer to the fact that this was a very fragile piece of living leather that had to be protected by technology that also acted as this surrogate body for this strange organ that we are growing, and this organ was basically a tiny jacket. So we exhibited initially in a textile and fashion show in Perth, and then in some other places around the world, and then in 2008 we were invited to show this work in MoMA in New York. It was a design show, a very optimistic design show. It was called Design and Elastic Mind, and the idea there was that if you get designers and artists to work with scientists and technologists, they'll be able to solve the wicked problems of the world. There were about 250 works in this show. Only one of them was alive or semi-living, and that was our victimless leather. The way we work is that we would, rather than travel with living biological materials from Australia, we would often make contact with the local laboratory and work with the scientists there, which is actually a really great intervention as well. In this case, we were able to make contact with some bioengineers in Columbia University, and we decided to move away from the original type of tissue that we use for the victimless leather, which was actually a mix of human and mouse cells that were co-cultured, that grow together, to working with mouse embryonic stem cells. What we didn't realize is that those mouse embryonic stem cells were growing so fast that about 5 weeks into the show, some of them sheared from this polymer structure and traveled throughout the system and clogged this life-sustaining mechanism or this surrogate body. There was like an amazing moment here where the life really literally grows out of control and chokes itself. We got a phone call from Paolo Antonelli, the curator, and said, like, what are we going to do? And we said, just turn it off. There's not much we can do about it. And Paolo Antonelli, the curator, rather than hiding it, like many other museums have done and will do, decided to go public with that. And you can imagine going in America, 2008, talking about how bad she felt about turning off the life support for a bunch of cells grown into a shape of a jacket as the pro-life, pro-choice debate was raging in America. Yeah, she was brave. And she basically was talking about kind of this, how bad she felt about it, and really transformed the whole conversation, not just regarding this piece, but in regard to much of the exhibition, because suddenly the discourse moved from what happens when you get artists and designers to help us fix the world to what kind of responsibilities we might have to exercise in regard to what we're doing, and especially when it comes to manipulating living biological materials. And as you can imagine, the media had a field day. Never thought that I'll have a headline in New York Times saying MoMA kills art, and it will be my own art that was killed. But it changed the discourse, and it changed the discourse from my perspective to the better. And here's kind of a little thing about the difference between art and design. Our designer friends came to us and said, "You failed. You didn't manage to keep your work alive for the duration of the show. It's a failed design work." And our artistic friends would come back and say, "You succeeded. You managed to generate the kind of conversation that was very needed in order for those kind of works to actually engage." Now, also I should add that there was nothing victimless about this leather. We're using a lot of animal-derived materials in the process of growing this. You need to use up to about 20% of what's called fetal calf serum, which is the blood plasma of an unborn cow. So the title was misleading, but many people took it on face value, including in this conference in 2018, that was a conference of startup companies meet investors in New York around the whole field of biofabricate, around this idea of using biological technologies for consumer products. And they decided to have a timeline of this whole new field of biofabrication and cellular agriculture. And to my surprise, they chose the victimless leather from 2004 as the trigger. And it kind of shocked me. I suppose many people would be really proud of something like that. That caused a huge existential crisis for me, because it meant that everything I've been working for as in kind of series of what I wanted to be seen as challenging cautionary tales became an instruction manual, and what propelled the very same thing that I said, "Hey, stop, we need to think about it," propelled it further, and using our work and basically appropriating it for this purpose. But that raises another issue, which is there was no invention in the traditional way of meaning of the word with this work. The technologies we're using, some of them were 100 years old, as I presented. Some of them are maybe 20, 25 years old. There was no technological breakthrough there. The breakthrough was that we as artists liberated the knowledge from the epistemological side of the biomedical world, and by that we opened up the floodgate for other people to exploit and extract value from. So I'll leave it there, but there's many other projects if you want to talk, but thank you so much.
Thank you so much. Thank you for such I could listen to you for hours, so it's a treat to get to hear you twice in, I think, about a week. So very, very grateful for that, and very grateful for all the diehards who've still stayed on towards the end. I'm also very thrilled to be here. I'm a great believer in string theory, and before I took the job at Wits, I was having very serious conversations about Leora, about coming to do my PhD with her. And in the string theory world, there is another me who is currently doing her PhD. So maybe it's that me that's actually talking here tonight and not the other me's. Many strange forms of light, indeed. I also, on a more serious note, don't think there's ever been a more urgent time to be discussing what light means and how we understand light at a time when genocide is being perpetuated around the globe. And I've lost a lot of faith in humanity. I'm asking a lot of questions. We're often told that as writers and artists, we bring humanity to things, and I don't really know if I want to bring humanity. I think I'd very often rather bring bacteria to things. Nonetheless, I wanted to situate my response quite firmly here in Johannesburg. In South Africa's very haunted history, under apartheid, the biological weapons program, which was named Project Coast and headed by Wouter Basson, aka Dr. Death, weaponized science against entire populations, producing toxins, fertility controls, and assassination tools. And it reminds us that laboratories are never innocent spaces. The same tools that can grow victimless leather can as easily engineer death. And I want to ask what ghosts of Project Coast linger in today's biotechnologies, in the tissue culture experiments that blur art and science. In Achille Mbembe's terms, we live in a country where many people within it are thrust into a condition he calls half-life, a zone where life and death interpenetrate, where bodies are reduced to spectral survival, and where zombified existence is created under a shadow of capital and violence. The living dead, the dying alive, every cell already contaminated by history. Bioart, semi-living sculptures echo this ambivalence, but are they utopian gestures or new forms of techno-violence, life commodified and suspended? African literature has long grappled with this terrain. Sunny Lavotanzi's Life and a Half imagines bodies that refuse to die, that return and again, even under regimes of terror. And this makes me think of Biko's words, it's better to die for an idea that will live than to live for an idea that will die. Amos Tutuola's Ghost Worlds in My Life in the Bush of Ghosts, where the dead far outnumber the living, insists that death is never a clean break. Weary, licking dreams of love across the hundreds of lives, a cellular love engineered to persist beyond the cut of history. Finally, Kojo Lang's Major Gentle and the Achimota Wars pushes this even further. It's set in a speculative 2020. This was written in the 60s, so the 2020 was far away back then, where the Achimotans fight wars not just over territory, but over futurity itself, so over the future and who owns and understands and controls concepts of the future. What they were defending their city against was, interestingly enough, South Africa's attempt to capture and commodify their qualitative futurity as a resource for its quantitative future. Crucially in the book, nature is itself conscripted into the battle. Rivers divert their course to drown invading armies. Crows pluck eyes out the eyes of mercenaries. Bullets fire their own guns. Moths and butterflies erupt from the body of an enemy commander. There's a kind of ecological insurgence, organic matter turned into bioweapon. And it's a radical inversion of Project Coast, where apartheid science sought to weaponize biology against Black life. Lang imagines a cosmos where the elements of Earth revolt against colonialism. These texts propose ontologies of the semi-living, haunted, resistant, sometimes ludicrous, always political. They remind us that what is at stake is not only the category of life in the laboratory, but the lived futures of those already consigned to half-life in the post-colony. So my provocation, Prof Catts, is this. Rather than seeing the poverty of language around life as a lack, perhaps it's a refusal. And I was very interested in you brought up a small point in your talk during the colloquium about biology's refusal to cooperate often with science, and I'm really intrigued by that. So as a way of life insisting it cannot be endlessly subdivided into biological, synthetic, virtual, perhaps the task is not to proliferate categories of life in the lab, but to learn from the half-lives outside the lab, from the mine, from the township, from Hillbrow just down the road, from the camps where survival itself is already a kind of bio-art. And perhaps following Lang, it is to imagine that the earth may itself possibly become insurgent, and that our experiment in the biology itself has insurgent possibilities. So yeah, I guess that's the essence of my provocation. I'd really be interested in you speaking about this idea of resistance within the lab and within biology, and also within your own art. I think that there's a wonderful way that, and I like it about your work, that there's an openness to it. Are they proposing this or are they critiquing it? And I'm curious to you to say, is that a willful dance? Do you have a stance? Are you willing to take one? And then a final another question that I feel really compelled to that links to this, but I just want to raise because it was a big point used on social media. The wonderful quote of yours, Prof Catts, no one wants to cross a speculative bridge. We want to cross real bridges. That was used a lot in advertising this event. And I was really struck by how often in Africa we are forced to cross speculative bridges instead of real ones. Across the Congo River, which has long been both a lifeline and a barrier, across the Limpopo, where migrants from Zimbabwe risk their lives to enter South Africa, across seas and rivers where survival depends on stepping into the unknown. If these crossings are already speculative acts, what does it mean to ask for a real bridge? And what kind of language do we need, not only for laboratory life, but for these lived crossings that are already speculative by necessity? I'll hand back to you, and then I believe we're going to open it up to you for your questions and provocation.
Thanks for making my life easy, complicated. So a couple of things kind of popped up. To do with your provocation, I think that the strength of art is when it's ambiguous rather than prescriptive or didactic. So in a sense, I like this tension where people are not really sure. And that's why obviously our work has been misappropriated in so many different ways. And because, as you mentioned, I'm acutely aware that life would resist. And a lot of the work that Jonathan and myself are dealing with deals with kind of a critique of human-centric approaches that are trying to assert control over systems that exist outside of our control, over complex, chaotic systems to some extent. So we don't want, so when we do a piece of work and we release it into the wild, put it in a gallery, and most people obviously are experiencing our work second or even third hand. They would either see a social media post, or they would read about it, or someone would tell them about it. The artwork literally gets a life of its own. And actually, I'm really interested in where this life is going. So the Victimus letter, I think it's the most successful in the sense of going places where I never really thought it's going to go. So following it is actually give me, I said it was an existential crisis, but also there was kind of this perverse pleasure of here, my kid, my baby, find its own way. He became a bloody conservative capitalist that is kind of engaging this most extractive of approaches towards life. And okay, it's alive in a sense, alive as a art of artwork that exists outside of my control. So I'm not one of those artists that try to overly control the narrative about work. I throw it, it's basically throwing a stone into a pond and let the ripple hit whatever they hit. That's the most I can do. Obviously, hopefully you understand I do have a position. So in our writings and obviously in our public talks, we tend to be a bit more explicit, but again, trying not to be overly prescriptive in regard to that. When it comes to violence, this is an interesting one. This is something that I've been pushing more and more as this whole area of bioart and biodesign kind of grew. I mentioned, I think yesterday, that our work started before the term was coined. Yeah, so I don't really see myself as bioartist. I'm sorry, but I'm an artist that works with biology, and I would stay like that. If people want to label me a bioartist, that's okay, but that's not really my interest. But as this field progresses, a lot of the discourse about it, and especially coming from very privileged, affluent people coming from very privileged, affluent background, would talk about their work as some kind of a collaborative work with the living forms that they were manipulating. They would talk about it in terms of this romantic relationship that they might have with those life forms that they forced to do the work for them. This notion that somehow this relationship is a mutualistic relationship where there's both sides are benefiting, I find problematic. So we started to talk about the notion, and this is a problematic approach as well, because we run the risk of flattening violence. But my take is that any attempt to assert one's control over a system that existed outside of their control is by definition an act of violence. This is the starting point. Your choice is the level of violence that you're willing to exercise towards the other. This idea, I've seen a philosopher, I won't mention her name, she was talking about it was a love letter to plants, and she was talking about rewilding, and she was talking about growing a garden. She never talked even once, and how much she loves the plants, but she not even once talked about weeding, killing plants in order for her plants to grow there because she prefers those, because she's asserting her human will over those systems. So violence is ever present in this field, and I think we need to acknowledge that. And then I think that's the starting point. Then we can strive towards the minimization of violence. But and again, many of my talks around here was talking also about this idea that what technology does, and especially biotechnology, and a lot of the work I've been doing lately is around trying to critique those new food systems that somehow promising the idea that we can exist outside of nature, that we can basically somehow separate humans from land in the name of sustainability and in the name of caring for the living others, while basically what they're doing, they're using more and more technology to obscure and hide the actual victims. They're creating a massive amount of what in economy we refer to as externalities, those costs that we cannot monetize, and therefore they're not in the loss and profit sheets. Yeah, but those are the real costs. Those are the costs that our kids are going to pay, and our grandkids, and the environment, and all of the species around that. Those are not costs that we measure. Those are exactly the kind of things that this technology is really good at obscuring and hiding, but not eliminating. And we need to be reminded that in order for us to exist, many other life forms pay the price for it. And giving at least an acknowledgement and thanks, I did the acknowledgement to country. In Australia, there's a debate. The Conservatives are, why do we need to welcome or acknowledge the land that we already live in? This acknowledgement is at least one step towards the understanding of the price that others are paying for your existence. And it's not just within an acknowledgement of the indigenous people of the land. We need to somehow acknowledge all of the life forms that pay the price for us to exist. And in VIAD, it's even more important because, as you said, dying for an idea that would live or living for an idea that would die. Many of the bioartists, I think, are living for ideas that are dying.
Well, firstly, thank you very, very much to Prof Catts for an incredibly thought-provoking, challenging, and inspiring set of ideas which you've put forward to us tonight. I think you've left us with a lot that we couldn't even begin to really unpack in such a short time. And I hope that there's going to be many other opportunities where we could further these incredible discussions.